we have like 1 minute before we actually need to start. On Friday, we'll have the review. The 9 o'clock class discussed this and said that they'd prefer to have like a lecture of like about 2/3 of it going over this last third of the course, like these are the things we talked about, and then maybe the last third of that being remember the whole course. Does that work for you all? Is this more or less what people are feeling? Works for us? Concerns? Other things people would like to do? Okay, you're like, yep, that's what we're doing. Great, okay, it does look like it's time to start now. So many things to go over today. But it's all fairly elegant, if that makes sense. Basically, there's one 2-minute video. If you understand the 2-minute video, you've got the chapter down. So, you saw this, I know you saw this, we're just going to quickly go over it. This was on Monday. We're in DNA, we're reading DNA, we're doing a couple things to get RNA made, to get RNA transcribed. I know you saw this. You have on the DNA, you have a promoter region that says, hey, there's a gene nearby here, you should probably go make it. There's some proteins that bind to this that help this RNA polymerase show up. RNA polymerase is going to show up and transcribe RNA. Fair enough? As this happens, you're going to be adding RNA nucleotides. This DNA, it always creeps me out when the mouse does that. Right? A little bit? It just, anyways. Fine. Stop doing that, mouse. You're going along, reading this DNA, building this RNA transcript. It does not destroy or use up the DNA at all. You can keep doing this over and over and over again. In true or false, it's a bad idea to like be driving right in front of a semi-truck and then slam on your brakes. Yeah, bad idea. Okay. Okay. With bacteria, they go along and they get to what's called the terminator, and they just stop as soon as they get there. For eukaryotic cells, kind of like how semi-trucks don't stop immediately, they'll get to they'll get something that's called the polyadenylation sequence. They get to a sequence that says, hey, this is the end. And 10 to 20 to 30 nucleotides later, it actually stops. So far, so good. It goes a little bit long. Okay, so that was to finish up through Monday stuff. Today, we're on to the punchline of the whole course, which is we're going to make proteins. Proteins have been appearing the whole time, right? Proteins have been acting as enzymes. Proteins have been being structural stuff. Proteins have been all over the place. And the idea of you have DNA, you make mRNA, from mRNA you make proteins, right? The idea we're solid. Yes? Great. So human cells make about 100,000 different types of functional proteins. Yeah? A lot. But the best guess puts this around 100,000. So any guesses how many genes people have? It's kind of a trick question. How many do you want to say based on this information? Back in the day, when people were doing the Human Genome Project, which was an amazing project, by the way, the biologists working on this knew you're going to need a gene to make a protein, right? And people were taking guesses about how many genes there were, and the guesses were all near this 100,000 number, because they knew you need a gene to make a protein, right? People would have bet money on this, right? Because obviously, right? The real number of genes, because the Human Genome Project, amazing, got it done, and when you count those genes, you end up with about 30,000 genes, which is a much smaller number than 100,000, right? And people were like, what? Right? And do you see why people were like, what? Because you need a gene to make a protein. What gives? Yes, and that blew people's minds. I'm going to repeat it just for the online people. Ben said the same gene can make different proteins. Yeah. So there's a special way of how this works. We're going to look at it. The key to all of this is after transcription, eukaryotic cells modify their mRNA. So bacterial prokaryotic cells, they just do their own thing. But enzymes in the nucleus are going to help modify this RNA that just was made. It's called pre-mRNA because it's not really left yet. What's going to happen are a couple things. It's going to modify the ends. So your cell is, the cell as a whole, is all excited about chewing up things that aren't being used. So if it sees some random string of RNA, it'll be like, ooh, tasty, and like pulled apart, right? So if you've gone to all the effort to make a piece of RNA, you maybe want the protein that this thing codes for, so you don't want your cell to tear it apart. So you put caps on the end. You put what's on the 5' end, you put what's called a 5' cap, and on the 3' end, it's called a poly A tail. And they're basically keeping enzymes from destroying the stuff, right? It's there, it's not going to get chewed to bits. You basically just make sure both ends are fine. It also is going to help this mRNA leave the nucleus, and the 5', this 5' cap is also going to help the ribosome find this thing. So basically, the first bit of processing that happens is just like, let's protect the ends so bad things don't happen. Fair enough? The next bit is where it gets cool. It really gets cool. This whole long thing is meant to represent this mRNA transcript that just got made, right? And it's been processed a little bit. It's got that 5' cap, and now has a poly A tail. Beautiful. There are parts of this thing that code for protein, right? And then there are parts in the middle that just are hanging around participating. What happens is, in this thing, there are long, long stretches that have non-coding regions in them. They're called intervening sequences because they intervene with the coding parts. So the intervening sequences are called introns. The part of this thing that actually gets expressed are called exons. They're actually expressed, they're able to find expression and become a protein. Okay? So you've got introns, which are intervening segments, and exons, which are expressed. RNA splicing is going to put these things together. It looks like this, and it's adorable. On this picture here, you've got the cap and the tail, great. These darker orangish colors. Okay, sorry. I'm okay on colors. I think I'm a little bit off, and I check when I claim different colors on things, because it's happened a couple times where I've been like, the blue thing or the green thing, and the class is like, no. Everyone's with me on orange? Yeah. These darker orange bits are the parts that are actually expressed. They know how to, like, build protein junk, okay? These lighter orange parts are the intervening sequences, they just did no. So, RNA splicing is going to do a couple things. It's going to cut and glue. And it can go cut, cut, glue, cut, cut, glue. And it can glue all these introns together. Which seems like a lot of extra work, right? Like, if you don't like that, why have it in the first place? Yeah? The cell knows how to cut and glue RNA, which means you can cut and glue it in different versions. It's called alternative RNA splicing, which is basically just saying you can have genes that make more than one protein. It looks like most genes actually end up making like 3 different proteins. It's the idea of, let's say you have a blueprint for a house, right? If you have to follow that blueprint exactly, one blueprint equals one house, right? If you can like modify that blueprint, one blueprint can equal more than one house, right? So you might have this blueprint that has like, this is how you make the kitchen, this is how you make bedroom, 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 bedroom. But if you're reading that and you're like, meh, I'm just gonna leave out that bedroom, you suddenly have a new house, a different kind of house than you could have built. This is what's gonna happen here. These different portions of proteins are typically called domains. So a domain might be something like for a blueprint, a domain would be like the kitchen or the roof, right? For a protein, it might be like this part binds to the membrane, or this part recognizes the signal. Yeah? So these different chunks that do things, you can put them together in different ways. It's called, what they're called domains. So that looks like this, where, let's say you have that bit of DNA up there, where you have exon 1, exon 2, and exon 3. They code for protein domains 1, 2, and 3. If you just read it like that, cut out the intervening sequences, and just read all those expressed bits, you end up with that protein, right? So we're seeing DNA here, but the RNA is actually where this is happening. If you cut out between exon 1 and exon 3, that means this exon 2 right here is treated like an intron, and you'd end up with a protein that's this green bit and then this purple bit, right? So you have a new protein, or a different kind of protein from the same information, but you've now done something different to it. There are lots of options. You could have this whole, all 3 of these domains, just from this one. You have this whole thing, you could have this and this, right? Where if you cut, do you want just these 2? Are we okay to cut the exon 2? We still cut out the first intron, right? But then we don't add exon 3 in. So you have these 2, you can have these 2, you could also have just these 2, cut out the middle one. You can have this by itself, or this by itself. The only thing you can't really do is you can't have it in the order something like yellow, purple, green, because you don't like get to actually rearrange, but you can't just cut and glue together in different ways. Fair enough? So you can end up with so many different proteins from the same, like, masterpiece of DNA. It's very nice. I don't know why it amuses me. Partially just because it blew everybody's mind. They're like, okay, we need to learn more about stuff. Great. So now you've gone to all this effort. It's been a lot of work for your little cell. You've made this pre-mRNA piece, and now if you're a bacteria buddy, you've just gone for it. But if you're in the eukaryotic cell, you've modified it, the ends have caps, the middle's been sorted out, everything's great. Yeah? Now you need to actually get to the punchline of the class, right? You need to make some proteins. This is going to be translation. The piece of RNA that you're reading to have it mean something, that is acting as your messenger from the DNA, right? It's called messenger RNA or mRNA because it has the message for how to build the protein. Yeah? The cell is going to read that messenger RNA, that mRNA, with the help of something called transfer RNA or tRNA, which is going to transfer amino acids around. DNA and RNA are made out of nucleic acids. Proteins are made out of amino acids, right? So tRNA is going to transfer amino acids around. The whole system of how this works is a little bit complicated, but the basic outline is very, very elegant. It's this. Yeah. We have a piece of messenger RNA that's being held by a ribosome. These transfer RNAs are going to find the codons. So the codons, remember those 3 nucleotide chunk that codes for specific amino acid. Transfer RNAs have things called anticodons, meaning if this is GGC, this is CCG, because it's the opposite, it matches. That matching allows this one to fit. If it were some other transfer RNA that has a different anticodon, if it were to try to come in here, it wouldn't fit, it would just bounce back off, right? So this matching lets things fit. There's a string of amino acids here. It's called a polypeptide chain. That string up there of all these amino acids are being held together by covalent bonds. This transfer RNA is holding an amino acid. It's going to come, and this little amino acid and this little amino acid are going to get covalently bound together, and the whole thing's going to just shift back along. We'll see this in more detail. As this is happening, once this one has passed on this string, this transfer RNA that just brought the new amino acid, this transfer RNA doesn't have the amino acid that's holding. It's empty, it goes away, and it's going to get activated. It's going to get an amino acid put back on it. Yeah? So far, so good? These little tRNAs, represented by holding different shapes, they're not identical. Each of them is going to have an anticodon on one end, and the anticodon is what's pairing with that complementary codon on the mRNA. tRNA, this transfer RNA, is made out of RNA. If it's flat, it looks like this, right? So you've got this long piece, this reasonably short piece of RNA. There at the top, there's a portion that can hold an amino acid. It can grab an amino acid. On this end, there's a portion that's the anticodon that matches with a codon of a specific type. This is flat, but people are good at doing this. If you imagine how it's folded over and then folded over, it becomes this shape. That's why this is folded. You have the anticodon, the amino acid attachment site. Your book draws it as this little blue squiggly thing, but that's what's happening there. To have this whole system work, you need 2 things to happen. You need to have this anticodon match with whatever amino acid you put on that end, right? Just like if you're putting labels on cans of food in some factory, you want to make sure that whatever's inside the can matches the label you put on it, right? Because you mess up refried beans and dog food one time, and you don't have a job, right? Okay, you need that to happen. You then, later, I think I have a slide up. Yes, I have a slide up. Then after you've done this, you need to have that tRNA anticodon match the mRNA codon that you're trying to match it to. The first bit, getting the right amino acid onto your tRNA, has this enzyme called synthetase, which is cool. It looks like this. You have your empty amino acid that has a specific anticodon on it. There's this enzyme called, this one's tyrosine tRNA synthetase. This one's doing tyrosine. Other synthetases will do other amino acids, all right? But this one's a certain shape, and that shape means the tRNA that has the anticodon for tyrosine, this AUA portion, that fits. It fits beautifully in that enzyme, okay? It can fit and it can be held. This enzyme also fits and can hold tyrosine, right? So tyrosine fits there. tRNA that's complementary, so tRNA that has the anticodon that matches for tyrosine also fits. They both show up there, and with a little bit of energy, you can attach that tyrosine amino acid to that tRNA. Fair enough? The reason these things work, well, you have, I don't know, if you have some other amino acid, you have a different enzyme that fits the other amino acid, and that enzyme also fits the anticodon that matches. Are we okay with that? Lots of different versions of this RNA synthetase, but each one is specific for the tRNA and the amino acid that are supposed to come together, right? Concerns about this? People are shaking their heads. That's a good thing, I think. Great. So this is what your book draws. This is more actually what it looks like. You've got the anticodon that fits here, so this thing is being held there. The amino acid fits nicely in this little cubbyhole and gets attached here. tRNAs that don't match here don't glom on. Amino acids that don't fit here don't come in. So you have the correct pairing of these things. Oh, right, and then it's released. Okay, so now you have your tRNAs activated. They're ready to go. You now need this to actually happen. Enter ribosomes. They're going to help the mRNA and the tRNA interact. If you have brain space for only one thing today, the first half of this is what you want to commit to memory. And this recording is by the cheerful lady. There are a couple different voices. This one's the cheerful one. Come on.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected.
and the remaining segments of RNA are reconnected.
Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
We're cool? These should be in your notes. Yeah. It's easier to see it in 3D animation than it is just to, see it flat on the page. But we're going to try. Here's our ribosome. You've got a couple different parts. You've got the E site for exit, the P site where you've got this polypeptide chain coming out, and you've got the A site, which is where you're going to bring the amino acid. Note the naming convention. Yeah. Speaking English is a help in biology. It just really is, because a lot of things got named in English. And yeah. But that's what we've got. We've got mRNA that's going to bind there as well. So here's our take-home picture. Right? So far, so good? All right, the P site's holding the growing polypeptide chain. The A site is where the tRNA that's carrying the new amino acids can be added, comes in. And the E site's the exit site. Right? So to get this to go, you have to start it. It's called initiation. You have to keep going. It's called elongation, keeping going. And you have to terminate with it, which is the end. All these steps are going to have some proteins that help make this happen. And you also need to pay for it with some ATP, too, a couple times. Oh, so many words. Words trying to describe this, mostly for your study. You've got your mRNA that's bound. You have that start codon, right? That start codon, that AUG, means go. That, you're going to have a tRNA bind to that. With a little bit of energy that will then pop the top of the ribosome on. So now you're all set. You now need, so you've started, this is the initiation part. You've started it, you now need to elongate it, make it longer. Here, we're just going to look at, all the words are going to describe what I'm trying to describe here. You have this situation going on here. The codon is recognized by that tRNA. right? So the anticodon and the codon match. If they don't match, the tRNA doesn't stick, right? It kind of try another one, matches, it sticks. Then it's going to take a little bit of energy to transfer that growing polypeptide chain, that string of what looks like a string of beads, makes a covalent bond between this one and this one. All my people, covalent bond between here and here, right? Then the whole thing is going to shift down, and that new transfer RNA that came in, as this whole thing shifts, is going to end up there. It's going to have this growing polypeptide chain, and this one that just a second ago was in the A site carrying the amino acid, now is in the P site carrying the whole growing polypeptide chain, and the process can repeat. So far, so good? The last bit is when you terminate this. So you keep growing, you keep growing, you keep growing, until you get to the stop signal on this. You want to end, right? Have you ever you're handing somebody something, and you think they've grabbed it, but they haven't? It's strange, disconcerting. You hand it to them, let go of it, and then just boom, it drops. Yeah. That's the basic mechanism for termination. So, once you get to the stop codon mRNA, you let something called a release factor in, and the release factor isn't holding amino acid. It's going to add water on accident. Well, not on accident, it's on purpose. It looks like this. This ribosome, mRNA, tRNA situation has been moving along. It's going great. When you get to the stop codon, there are several versions. Instead of having a tRNA that's holding an amino acid come, you have this release factor that matches. The release factor matches that codon. The release factor shows up. It is not holding an amino acid. Simplify it a little bit. It's basically holding a water molecule. Happy to help. It's there. The ribosome does what it's trying to do always and tries to hand this growing polypeptide chain to this thing that's in the next spot. And instead of grabbing onto amino acid, it grabs onto a water molecule, and now it's done because nothing's holding onto it. So the polypeptide chain floats away. The tRNA that was just there, scoots along. It's gone, it's off. And then the whole system just disassembles. Fair enough? You see how you're going along, reading this, adding bits, adding amino acids, adding amino acids, adding amino acids, until you get to the stop codon. You don't, you just let the thing go. Great. Concerns about this? I feel like I'm just whipping through things very quickly. But people are nodding, so that's a good sign. Have you ever, this is more relevant for people perhaps who have a number of siblings, but this happens with friends too. You both want to read the same comic book or whatever at the same time. Ever happened? Have you ever tried the exciting thing that seems like a good idea but doesn't work very well, where like you read and you're both reading at the same time but with like different pages, right? So you're reading here and you have the page up and the other person's reading the next page. Doesn't work well. My siblings and I tried it a number of times and we never really got it to work, but we tried it, right? We were all reading at the same time. Ribosomes are ahead of where we are with that idea. They've figured out how to read the same piece of mRNA in different proportions. So whatever cellular event has happened that makes your cell want some protein, you've gone to a lot of molecular trouble to read the DNA, make mRNA, and yeah, you can make lots of different mRNA. You can make lots of copies of mRNA, but each copy that you make is expensive. So you make this mRNA, you've gone to the trouble of processing it appropriately, and now it's outside inside the cell, in the cytoplasm of the cell. What you want to do is you want to use a lot of ribosomes on the same piece of mRNA. You can read this piece of mRNA over and over and over. It looks like this. You've got this mRNA here. The ribosome comes in. You've got, in this position, you've got 4 other ones reading the same thing. You can see it right here. A piece of mRNA along the whole thing. All these mRNAs are reading it, and they're growing proteins. Once the ribosome gets to the end of it, it's going to unattach. You can keep putting new ones on. You can have one piece of mRNA read over and over and over and over and over at the same time. This makes the system way faster to build proteins. We're good? Questions? Ooh. Okay. Once this ribosome gets to the end of your piece of mRNA, you have this whole polypeptide string, this whole string of amino acids. You have to fold it to actually make it be a protein, right? But once it's folded and processed, then you have a beautiful protein. You did all that you wanted to in biology, you're there. This is the picture that has everything happening at the same time. You have transcription, right, where you're going from DNA to make RNA. You have some RNA processing. There's nucleus. Because there's a nucleus in the RNA processing, we know we're in a eukaryotic cell here. That processed bit of mRNA comes out. The ribosome attaches to it. tRNAs with their anticodons bind to the codons. Translation happens here, right? This is translation. Reading information in nucleic acid and building something in amino acids. And then we've got our amino acid activation. That's just where you put the amino acid onto that tRNA. Questions? Do your neighbors look confused? Nope. Great. We have a little bit of time for then mutations. Yeah. These are the things to think about. We're now going to talk about mutations because this is a good spot to end the course. In Hollywood, tell me about mutations. Right, like I saw Spider-Man a long time ago, but like you've got this like mutant spider that like rah, right? Depending on the version of Spider-Man, just for the record, what makes Spider-Man fancy or special in different iterations of Spider-Man depend on what technology is new at the time. So old Spider-Man was bit by a radioactive spider. New Spider-Man has been bit by like a transgenic spider. Anyways, just depends on what we're worried about at the time. Okay, regardless, mutations often are like, whoa, something weird, like everything was fine, and then cosmic space blast, and now you can shoot lasers out of your eyeballs, right? Mutations in this class are less exciting. Like they're nice or not, but they're not like shoot lasers out of your eyeballs. So all a mutation is a change in the genetic material of a cell or a virus. All right? You have mutations happening to you all the time. Mutations that happen to your germline cells can actually get passed on to the next generation. But other mutations that just happen because you were out sunbathing too long and you got kind of sunburnt and cooked, you know, it happens. All right? If you change the DNA on the template strand, you can end up changing what the protein is that's made from this, right? Something like this. Normally, when you're trying to build a blood cell, you need a protein called hemoglobin, and the DNA that does this has this pattern on the template strand that's CTC, right? That after you transcribe it, you end up with this GAG on the mRNA, which then normal hemoglobin. You read this GAG that codes for what? Glutamate. And you have that amino acid that fits there. So far, so good. If you have a mutation there in your DNA that instead of saying CTC, it says CAC, when you make that mRNA, it's going to be GUG. Just one change, but that change says to grab a different amino acid, right? So when that mRNA is being translated, the tRNA is doing its job. It sees that codon, the anticodon that matches that comes in, and you put a different amino acid on it. Fair enough? So that mutation has switched what the protein's going to look like, and in this case, it's been bad, right? Okay. Mutations can happen in a number of different ways. There are two main categories of these. And we're talking like fairly small scale mutations. If there's like huge mutations, there are other issues going on. Point mutations can happen in two different versions. You can either substitute one nucleic acid for the other, or you can just add extra bits or take out bits. We're going to see the substitution part first. Okay, so substitution. Once upon a time, and it's gone down as family legend. You know those stories that, like, make into family legend, become even better than the original? My sister, I know we were teenagers, she's all excited. She's making this new recipe, and she's got this cool cookbook, and she's a really good cook. But we go and we eat it, and there's this like, new recipe, huh? She's like, yeah, it's terrible. I hate this recipe. It's awful. I'm never using it again. We're like, what is it? She's like, it's Chinese broccoli cashew chicken. And we're like, Manita, this is beef. She's like, I know, we didn't have chicken, so I substituted beef. Also, these are peanuts. I know, we didn't have cashews, so I substituted, peanuts. Also, should there onions in this? Yes, we didn't have broccoli, so put onions. We're like, okay, so like, it's not really the recipe. You just made too many. You can substitute one or two things. But when you're substituting like peanut butter for soy sauce, like, you're gone, right? Okay. Mutations have a similar idea happening sometimes. Some substitutions, if you substitute garlic powder for onion powder, you're probably fine. If you substitute milk for tuna fish, something weird's happened, right? Okay. Sorry, it's not always food, but food is the easiest concrete. Okay, I realize I've got notes about this. This is why. There was an architecture analogy earlier. Okay, so substitutions, you're substituting one thing for the other. Silent mutations are great. I mean, they're not actually good or bad. They just don't cause a problem. They have no effect on what amino acid gets changed. So if the codon CCG and the codon CCC both code for the exact same amino acid because they're redundant, having a mutation that goes from CCG to CCC, you don't even notice it. Fair enough? Like, makes absolutely no difference in the protein that's built. Also, if you just have a substitution in like an intron, you're not going to notice that either. Those are called silent mutations. No effect on the protein. Like, it just doesn't. Like, yes, something's changed, but meh. Missense mutations are scarier in that they're still going to code for an amino acid, but not the right amino acid. This is a missense mutation. Codes for an amino acid, but it's not the one you're looking for. Theoretically, sometimes your protein might get better at doing whatever it's doing, but usually it gets worse. I mean, it's not usually. But basically, it just changes the thing, and often that can be a problem. And then, so, silent mutations have zero effect. Missense mutations, sometimes the protein still works, sometimes the protein works less well. Nonsense mutations are bad. Nonsense mutations change the amino acid codon from saying get an amino acid to a codon that says stop. So this isn't a huge deal if this is the very, very, very last amino acid you're putting on this growing polypeptide chain. But if, like, you change codon number 2 to say stop, you just don't have a protein because you started building it, but you just didn't get that far. It just ended. We're okay with that? Nonsense mutations nearly always make a non-functional protein. Some very, very, very rare times, you might have it be far enough near the end, that's not a problem, but usually you just shut this whole thing down, right? So substitutions are a problem, or at least they change what proteins you're going to make. Want to know something that's even more problematic? Just cutting chunks out, or adding additional things in that you're not supposed to. Insertions or deletions are where you add or subtract nucleotides in the middle of a gene. They usually cause bigger concerns because you shift the reading frame, right? So if you're reading 1, 2, 3, 1, 2, 3, 1, 2, 3, and you add one nucleotide in there, you're going to be off one on every single thing. This has happened when you've done tests. You've done those little Scantron bubble sheets, right? A substitution error is where you make an error on which circle you're bubbling, right? You meant to bubble A, but you meant to bubble B. That can be a problem, right? But it's not, like, disastrous. What's disastrous if you're 1 off on your bubble sheet, right? So you're marking 1 when you should be marking 2, you're marking 2 when you should be marking 3, and you're 1 off in every single thing. Yeah. Anybody ever done that? Yeah. You're like, no! Now I'm wrong for the wrong reasons because I'm just off, right? It's called a frame shift mutation that can happen with either adding or subtracting. Honestly, adding or subtracting 3 nucleotides is a better deal than adding or subtracting 1 or 2, because you're at least on the same reading frame. Yeah. With that, just the caveat, mutations, Hollywood aside, don't have to be bad. Typically, the way whatever, I don't know, if we're like looking at chipmunks, typically a chipmunk, like, it's doing fine as a chipmunk. If there's been a mutation, it doesn't usually make things better, but it sometimes can. Most mutations actually don't seem to make much difference. Some mutations can be really, really disastrous because you just break the protein. All right. Thoughts? Concerns? Comics? Yeah!
So, like, are these mutations responsible for evolution, or is that something?
Yes. Amongst other things, but yeah, so, yeah. In something like bacteria, let's do bacteria really fast. Ooh, how are we in time? Ooh, great, yeah. So, in bacteria, you've got, bacteria have way more mutations than we do. They're not as careful proofreading their DNA. You have some mutation. A lot of them don't make anything better, but some of them, you make a protein that's less likely to respond to antibiotics. So you've got your little bacteria buddies. They're living there. There's just some random fuzz, right? There's just some change in the DNA just because it happens. You go and you throw antibiotics on it. You kill all the ones except the ones that have the slightly better DNA to do this. And those are the ones that have a whole bunch of little bacteria. And there's random jiggling in there's some mutations. And you do it again, you keep picking for the ones that have the mutations you want. Yes. This has happened with, like, plants are another way to see it. You know, like, cabbage and Brussels sprouts, you can see, like, some similarity here. There's slight differences that long enough you can push them apart. But yeah, no, mutations are the original genetic variation. And then different individuals who have now different alleles because of different mutations will combine in different ways. But yeah. Yeah, so mutations don't have to be, like, terrible bad. But there are a lot of bacteria who have mutations that just, like, instantly kill the bacteria. For every little bacteria that has a mutation that's like, yay, I'm invincible. Yeah, you're basically playing the genetic lottery. Yeah. Other questions? Sweet. That's where we're at. I'll see you on Friday. You do not have a quiz on Friday, but the test will open.